for Module 4 in the course, marking almost 1/3 of the course. I'm pretty excited as we start to dive more into bioinformatics. I think this is the stage at which we are going to learn more about technologies and start to see what the data looks like as it comes from some of the machines. Today, the topic is the on sequencing, part 1. I'm pretty excited to discuss this topic. Very interesting ideas that we're going to highlight, as hopefully you will see today. So I hope everyone is enjoying the course. I know you have been working hard on assignments, quizzes, but so far I see everyone is doing very well. I'm pretty happy with the progress of all the students. And yeah, let's stay excited and a little bit more patience until we get into a stage where, you start maybe a little bit of coding activities and have more in action with some challenging problems and data handling. So this is bioinformatics. It's never one type of subject. It is interdisciplinary, so we cover some molecular biology. It has been dense, but I think very important. And now we're going to move to a little bit of technology side, and eventually we're going to see some of the outputs that comes from some of the machines. So we're going to look into today some interesting topics, such as Sanger sequencing. Now, this is a big thing in history of sequencing. The idea is now we know that there has been DNA, ideas and thoughts of how it looked like, the double helix, all of these findings. We understand this, the molecules, the directionality of the DNA, the bonds, the energy bonds, and so on and so forth. How can we read that information? We, of course, highlighted interesting topics on fluorescence and how these ideas can be used to screen and find some information, whether it's DNA sequencing or gene expression. So what was like one of the breakthroughs to be able to read the sequencing, marking this huge project that is called the Human Genome Project, that took roughly 10 to 10 years, a little bit more, to really release the first human genome. Nowadays, it takes maybe a few hours, but it was such a big breakthrough. Microarrays that's great technology, and that's going to take us into how we can find about gene expression, and then we'll move on into next generation sequencing. So some of the concepts we'll discuss here are not necessarily what's being used as of now or nowadays. Things are being developed all the time, but again, they are the core. They are the starting point of many of the things happening. So hopefully today we'll go through some of the concepts, get you familiarized with sequencing, Sanger sequencing process in particular. We want to study the sequencing process. I'm going to ask a couple of questions about, what do you think would be the limitation and so on and so forth. Yeah, treasure hunt activity, we're going to come to that. And we'll see how these ideas of fluorophores are going to be key for establishing the principle of Sanger sequencing. And we're going to highlight the Human Genome Project. It is a piece of history releasing the DNA language, this DNA molecule, which is, the code of life, if you want to give it a short acronym or summary. And then we will move on into microarray technologies, discuss a few steps there and different types. And we will see some of the formulas, if those who would like to see a little bit of information into how we get some data and can get some numbers. And then we'll move into next generation sequencing. So again, this is the area of bioinformatics. It is this point where we have intersection of biology, technology, and I would say the future. I'm not just excited for this topic and work on it actively, and happy to teach courses related to it, just because it's just a new interesting area. Bioinformatics is really the big thing, okay? And personally, I see it as where it brings lots of skills together and give me the chance to learn about many of the things at very important depth. At the same time, I see how much as humanity we are struggling with complex diseases, and understanding DNA and unlocking the mysterious of complex diseases through sequencing technologies is something very instrumental in shaping what we have today in terms of modern biology and medicine. So as we explore pioneering Sanger sequencing that led to the foundation of what? Decoding the DNA, and then discuss this monumental contribution to the Human Genome Project, which mapped the entire human genetic makeup. And so after that, as mentioned earlier, we'll explore the microarrays, a technology that gave us the chance to see gene expression in action. So all of these topics are really important, not just for scientists in a lab because they want to run some experiments. It's for all of us. This is about indeed understanding at the molecular level how things work, and who we are at that level, and how can we take advantage of this knowledge for key things. That is our human health, tackling diseases, and also trying to, with this area of bioinformatics, one of the interesting things, you keep raising questions, right? It is this interesting field that we keep asking questions. So we want to address fundamental questions and try to, about our existence, about our own mechanisms, and how can we improve our life. So here we go. We're going to start with the topic on Sanger sequencing. And of course, this is attributed to Fred Sanger, who is a British biochemist, born in 1918. He passed away in 2013. Now, he's renowned scientist. He made pioneering work in the field of molecular biology, especially developing the DNA sequencing techniques. And Sanger sequencing is made Sanger sequencing is named to show attribution to Fred Sanger, which is going to learn about it today. One thing interesting about Sanger is that he's one of the very few individuals that have won Nobel Prize in chemistry twice. What breakthroughs this person made in the history of chemistry, and bioinformatics, and so on and so forth. Some of the key contribution included amino acid sequencing. So it's the idea of developing a method for sequencing amino acids and proteins. So Fred Sanger successfully sequenced the hormone insulin, and which demonstrated that proteins have a defined chemical composition. A very important major contribution was the development was called the Sanger method for sequencing DNA. That is the groundbreaking technique that scientists use to read our genetic code. And this was instrumental for, again, the Human Genome Project. So he got these two Nobel Prizes, one I believe in 1958 and one in 1980 when he was awarded the second one as well. So he had, of course, shown a profound impact in the fields of biotechnology, medicine, genetics. So let's learn about the Sanger technology, where The coming slides that will show, or what you're going to see is kind of animation. So we'll go through the steps, kind of skim through the process. And you already have some of the principles there, but I'm going to highlight on specific ideas, and then I'm going to show you as well some video to illustrate that in the best possible way. And along the way, you'll have maybe a couple of questions. So we need to start with what? With sample. Remember experimental design now. If I'm interested to read the DNA and sequence the DNA, which is what is sequencing the DNA, is actually reading the DNA sequence and knowing what is the order of nucleotides that we have. So we start with the sample, and then from that sample that we can get it from a human, we need to isolate the DNA. So there is a process some of you may be aware of as biotechnicians, how to take a sample and then start to apply certain kit to get the DNA out of that. And now you probably understand that depending on the goal of our experiment, we may be working on isolating DNA, or maybe we may be working on isolating mRNA, okay? So here's one, I think, nice, interesting question for our treasure hunt activity. Why would we be interested in mRNA? Or let me rephrase this in isolating mRNA. Or let me rephrase this. When do you think we are going to isolate DNA, and when do you think we are going to isolate mRNA? Okay, and this would highlight understanding of what we have been doing so far. In other words, you can just explain the difference, why we are isolating DNA or RNA, and why does it matter. Okay, so as we do this isolation of DNA, we need to apply some heat. Now we understand why do we use heating to denature the DNA, the double helix, because we want to read the DNA. We cannot read it as it comes in double helix shape. We need to unwrap these two strands, and then start taking one strand and focus on that. So there is going to be a source of energy and heat where we can separate, denature, what's called denaturation, and at specific temperatures we can get to see the DNA strands, which so far would seem, I believe, a typical process. If I remind you about polymerase chain reaction, there are similar steps, and these are in general the steps that we expect to have when we are working with applying certain technologies. Now, we, as per the standard, and this is like originally how the Sanger technology worked. Okay, so as I present Sanger sequencing, I'm going to show you how it worked originally, and they will talk about high throughput way of doing it. The reason I'm highlighting this here, because what you see here is that we have 4 tubes, and in fact there were 4 tubes in the beginning. Okay, in the beginning there were 4 tubes, because each one is gonna tackle specific nucleotide, specific color, and specific scanning. Now this might cause a confusion for some of you, because all what we discussed earlier is, oh, we can handle this through 1 tube. Indeed, this is going to be the case with the recent development, but at the beginning, there were 4 tubes for this experiment. Now, what you see here is that once we have the DNA, so the DNA sample is going to be injected in each of these tubes. So there will be some DNA strands, and they will be common between these tubes. This is one thing. Another thing that will be part of the process the primers. Okay, the primers are designed to anneal to the beginning of the region to be sequenced. So this is the region to be sequenced, so primers are designed to start here. Now my second treasure hunt question is, why do we need primers? It's something we discussed before in one of the lectures. So why do we need primers here? So once we have primers, we can also put the primers in the different tubes. Now, this is for illustration and for simplicity. Otherwise, primers are not going to attach to DNA before they are put in the tubes. No, DNA will be here, sample in all of those, and then added would be the primers in all of those, because to have the primers bind, there should be some specific temperature, okay? Specific temperature is going to be allowed. That is going to allow the primer to anneal to the complementary sequence of DNA. Okay, so at this stage, for illustration purposes, we have DNA in all of those. We have primers in all of those. We're going to have nucleotides. Now, what are those nucleotides? Okay, and it is my third treasure hunt question. What are those nucleotides? Okay, I'm going to give you a moment before I jump into this. Think about those, because those are not just normal nucleotides. Those links to our last lecture that we started discussing, right? Each one is going to have a specific design where one fluorescence molecule is attached to it with a specific color indicating that specific nucleotide. What are we talking about? So share your answer, and they are DNTPs, okay? We call them DNTPs. We're going to come to this discussion because this is going to be core for the Sanger sequencing. So now DNTPs will be also put in every single tube, okay? Finally, Taq polymerase. Taq polymerase would also deserve, would also certainly deserve a question, a treasure hunt question, which we have looked into before. So what is special about Taq polymerase? We know that it is a DNA polymerase. It is a kind of enzyme, and that was derived from bacterium called Thermus aquaticus, right? And what is special about this polymerase, okay? And why do we need it, okay? So anyway, it is well known for its role that we discussed in polymerase chain reaction. We need a DNA polymerase. It is a kind of DNA polymerase. Well, why we're using Taq polymerase? This is the question for the treasure hunt, something we discussed in detail earlier. So Taq polymerase is going to be also injected in all of these tubes, which now, as you understand, we have DNAs, we have nucleotides, we have primers. Taq polymerase can now bind to these primers and start getting these nucleotides, binding them in order so we are able to construct the complementary strand of fluorescence, which we can then subject into certain technology and see what is coming out from this. However, as of now, you may directly jump into, we are going to shoot this with some laser and be able to read one by one. It links to what we discussed before in the previous lecture. However, the Sanger technology in the beginning was not built using that. It was built using, or Sanger sequencing, 4 tubes. Now, at this stage, I hope you settle with this. We have DNA in all of them. We have primers in all of them. We have all these nucleotides are going to go into all of them, okay? It's not like one tube at this stage is going to have one specific DNTPs. No, all of them are going to go into all, and then Taq polymerase into all. So at this stage, there's nothing that makes a single tube with those 4 tubes unique. Now, here's what is going to, here where we jump into when we are going to have each of these 4 tubes to be different, okay, and have a unique composition, okay? So what do we see here? This is the DNTPs that we have, the deoxynucleotide DNTP, okay? There is a hydroxyl group at this bond, which would allow it to go and bind for the next nucleotide. We know hydroxyl group, this is at 3' position. This is 1', 2', right? So this position can link to another phosphate group, right? This is how the backbone is built. This is the sugar part, this is the phosphate part, and then here we'd have a nitrogenous base, which is ACGT. So DNTP is having a hydroxyl group here, which means of one oxygen and one hydrogen, we call it hydroxyl group, that can bind to the next phosphate group, okay, of another nucleotide, okay. So that is what is in all tubes. Now there is another type of molecule that is called a dideoxynucleotide triophosphate. Dideoxynucleotide DDNTP. So this is DNTP and this is DNTP. Okay. Now, what is the difference here? You see it directly. They are exactly the same thing, except, and they will have fluorophore colors. Everything is fine. Everything is designed as per our need to have differentiate them in colors. The only difference here is that instead of having hydroxyl group, we have a hydrogen. And what is that change is gonna do? Okay, DDNTP is incorporated instead of a regular DNTP. This one is incorporated instead in the design of the experiment, okay, instead of the DNTP, because it lacks a 3' hydroxyl group. What that means is that it's going to terminate the DNA chain. So no further elongation, no further extension. So it is the process, I'm in the process of appending different nucleotides. I'm going to change the color, okay, so that you understand each one is going to have its own color. Oops, okay, just bear with me, okay, G and T, doesn't matter, and you keep going, okay, you keep going, yet those are DNTPs, okay, yet the moment we have something like, let me use this color again, C, which is not the DNTP, see, it's not DCTP, okay, it is DDCDB. I'm gonna do something extra here. It's gonna be kind of bolded, okay, bolded. I'm gonna bold it more, okay. Now, as this happens to come and be attached to the complementary strand of DNA, as the DNA polymer is going to work, is going to block any addition. So the whole thing is going to stop. There's no more elongation. So this interesting idea, which actually Fritz Sanger asked, what if I change the 3' here and I remove the oxygen group? Then I'm going to block it from enabling the next nucleotide end chain to come and bind to it through the phosphate group. We block that reaction. So what happens then is I'm going to end up with some read here. Okay. Now, here it might get tricky, but we're going to repeat this a couple of times so you understand it. But all I want you to see now is that we have now DNTP and DDNTP. And what is the difference? DDNTP does not allow elongation anymore. Now, for the sake of this difference, okay, the DNTPs, so let's say we have one for A, C, G, T, each one is going to be injected to one tube. At this stage, therefore, each tube would be different, okay, would be different, okay. At this stage, each one of these tubes is going to be different, meaning it's going to have some unique color at this stage. Well, the unique color is not coming from the DNTPs, it's going to come from the DDNTP. Let's see the process. Okay, and so as I said, we will have whatever I've drawn there as bolded. You can refer to it as CDD, TDD, GDD, or ADD, or DDGTP. Each tube now is going to be a different color, different representation, and so each one of these tubes is going to have a unique DDNTP, meaning that it's going to have a unique color once we start scanning this and moving into it. Now, what do you see here is something I want to illustrate again. Okay, now perhaps I would do this here. Okay, and I want to get you into this. Now, in one tube, let's say that we have tube 1 in this case. So I'm dealing with one of the tubes. And imagine that this was for DD. This is where we had DDATP. Okay, DDATP. So we have adenine. Now, what does it mean that we have DDATP? It means that adenine is going to mark the termination. Okay, it's going to mark the termination. Yet one of the things that I want to bring into attention again, before diving into this, is that in Sanger sequencing, the process starts with the template preparation. Template means, okay, let me write this here. Let me have a quick review here, okay, quick review of the process. And I'm going to go back to what is happening in that specific tube. Okay, let's have a quick review of the process. There is template preparation. Template preparation. Template preparation is a DNA fragment to be sequenced, okay? And then there is going to be a primer that is going to be designed. A part of this, I can also add to this, plus primer design, okay? So this is one step. Another step is reaction step. Reaction step, okay? So in this step, the mixture is prepared containing the DNA template, the primer, Taq polymerase, normal DNTPs, and the specially dyed labeled the terminating dyed deoxynucleotide triphosphate, which is DDNTPs. So all of those are the mixture, okay, the reaction mixture. Third step, so let's say this is 1, 2, 3, denaturation, okay. This is very important step now. Why? Because the reaction mixture that we have is going to be heated to high temperature. And this is usually something between 94 degrees to 98 degrees, okay? That's going to denature the DNA, separating the two strands. This is one step. So we are heating here. And there's fourth step is going to be annealing, okay? So now in annealing stage, what do we want to anneal? The primer to the complementary sequence. So we lower down the temperature, okay? So now we have the primer. What is next? What do you expect is going to be next? Okay, answer this question for me in the treasure hunt activity. What do you think is going to be the fifth step at this stage? And again, there is no wrong or right answer. All I'm looking for is some keywords that we can, make sure that you are going through, okay? And I want to mention something about the treasure hunt. I'm extremely happy with the activity of some of the students in the class. And I hope this is, making good interaction, that where you confirm that you have been watching the lecture and then also enjoy it. You get some extra credit. Not only this, I see the names who are contributing, doing well. I'm very positive with those students. If there's any chance to help throughout the course, of course, I'm going to dedicate that. So anyway, it's not only bonus, it's all support that I can give for those students because they are watching the lectures, they are patient, and they are writing down the questions. And, it's really one of the emails or messages I get in the chat that makes me very happy is about the treasure hunt questions. And I was not expecting it's going to be successful. It is very successful. It is very successful. There are a group, I'd say 1/3 so far for the class, 1/3, one of the class, very active with this activity, which is great. So thanks to all of you who has been working hard. So anyway, we are at the annealing stage now. What's going to be the next one? Okay, so it is going to be extension. Now, if you would say it's replication, whatever is going to be fine. It is the step where Taq DNA polymerase is going to work, okay? It's going to extend the primer by adding nucleotides. So whatever term you use, just to explain that at this stage, nucleotides are going to be added from the 3' end toward the 5', right? Let's start from the 3' end. Now, this is going to keep happening until the DNTP is going to be attached. And this is what's called a chain termination, okay? Yeah, let me put chain termination. Now, some of you may start asking, oh, we're a little bit confused, like, because I got a little bit confused when first studying this, is chain termination. Chain termination, when is going to terminate? Like when we're going to pick up the machine, is there a way where we pick up that DD in TP and then say terminate now? No, this happens randomly. So the termination may happen after one nucleotide, one D in TP, DD in TP and D in TP. After one D in TP is attached through extension, so maybe one, maybe a bunch of them, and then this comes. Remember, there is a mixture. There's no control in this mixture and whatsoever what happens in that stage, okay? Now, the reason here, I'm going to clarify a little more is step 7, which is very important step, amplification. You should remember this also from the previous lecture. Amplification basically is going to say steps 3 to 6 are going to be repeated. Steps 3 to 6 are going to be repeated, denaturation, annealing, and so on and so forth. This is going to create a set of copies of the DNA, okay? Which means in one tube at this stage, if I'm going to look into one tube at this stage, sorry guys, we will have sequences, okay, of different length. So we could have something like ATCG, okay, and okay, let's see, say C here, and they will have a TD and TB, so ADD. Let's mark it as ADD. So this is going to mark that there's no more extension. Now we repeat the process. We get another DNA strand in this same tube, okay? And so it could be ATG, CCC, ATC, and then we have now ADD again. So it's going to stop, okay? Nothing more, okay? We could have ATG, okay? So I'm going to mark the adenine here. It's going to mark the stop. And then we could have ATG, CC, CATC, okay? GAADD, okay? So we're going to mark the ending here. The reason I'm showing you this, which is going to be all part of one tube, okay? All part of one tube, which means in one tube, we're going to end up, because of this amplification process here and the repeat of these steps, what we're going to end up in every single tube is list of reads, okay? List of reads based on the DNA polymerase reaction and the generation of the set and extension of a new nucleotides. We're going to end up with list of reads because adenine of DD and TP stop the reaction once at one specific position. And again, as I said, we don't have really control. We don't have control, guys, here on where to add this. It's going to happen randomly, which is going to raise some other question, okay? I think I'll be interested in knowing another question, okay? Why? Okay, we understand now because of applying the Sanger way of technology, in each tube we will end up with varying lengths of sequence, DNA fragments. We'll have varying lengths of DNA fragments, okay? Now, is that something good or bad, okay? So I'm gonna ask you this question, give you a moment to think about it. at this stage, do you think like having DNA fragments of varying length is something good or not good? Okay. So just to give you a principle, the idea of the technology, this is the principle to work here because it's going to next be help us to read these DDTPs. But for now, having these multiple fragments, which is based coming from the amplification process, guarantees for us some level of comprehensive information. Okay, because repeating and having more fragments of varying length kind of ensures that every position along the DNA sequence is sampled and identified. Again, multiple fragments that terminate at the same nucleotide provide redundancy, which might be perceived as an extra accuracy, okay? And the varying length allows us also for high resolution and identification of each nucleotide, and so on and so forth. Complete sequence. Now, one can come and say, well, if I take the DNA as one strand and one piece and try to read it as one list of nucleotides, then everyone is happy, right? And I don't need any of these technologies. So let me remind you here that this is now what we're going to look into once we come to next generation sequencing. Okay, it makes sense to ask all of these questions. But again, we need to understand that there has been technology development through the years. What may seem now for us intuitive or a logical step or a complication, extra complication, this is just because of the fact of the technology development. Nowadays, still, what we're going to look into once we come to the sequence and learn some terms, the sequence length, sequence depth, and so on and so forth. One of the principal ideas here is that new technologies are trying to achieve longer reads. But given what we are looking at here now, without deviating into another discussion, the fact that we have different length and variation is given that these DD-NTPs are being introduced for a specific reason and goal. So nothing wrong with that because it's going to give us what we are looking for, which is reading the nucleotides. Okay. The other thing is that the technology and the whole development and the protocol wouldn't be easy to take and give a machine one full sequence of DNA and try to read it. And it may struggle with some accuracy at that stage or giving whatever technology was there. But nowadays, we should expect more efforts on that side. And in fact, there has been good, quiet developments in that. So that is why we see now in each of these tubes, we have sequences of different length. Okay. Now, giving one of those, like thiamine, for example, if I'm looking at DD TP of thiamine, this is to indicate for us that this is a stochastic process. It is random. That is why we end up with varying length of DNAs, okay, fragments. So I hope you see the point. So now we have everything. What is next in this process? Okay, so that is the question. What's going to be the next step in this whole process? Okay. So again, after things, if we were going to look into where I stopped there, I said 7. Let me change the color here to remind you that the same steps that we're looking at there. So we reached up to step 7, where I discussed amplification. Okay, amplification. So far, hopefully you see the process of Sanger sequencing. Next, okay, 8. Step 8 is going to be separation and detection. We are at this step right now, okay? And after that will become data analysis. That is something we're not focusing on in this presentation or in this lecture. So separation detection. The DNA fragments are separated by size using what's called specific medium. We're going to discuss 2 concepts related to that, okay? One of them is called gel, So yes. So separation and detection can happen through gel electrophoresis, okay, good. Make sure we have the right spelling. Looking at my notes. And the other thing is capillary electrophoresis, okay? Let's put this here for short, okay? So we have two different ways to do the separation and detection. We're going to look into this at first, and then we will refer to this as well, because this is the one that is going to help us to achieve high throughput, okay? High throughput. And it's the one that can be considered the more state of the art. But in principle, they share interesting ideas, okay? So the gel electrophoresis, for example, it is a gel made of what is called polyacrylamide, and this acts as a molecular sieve. This is kind of the medium that we are going to put into the solution or the mixture that we have from the 4 tubes. Remember the 4 tubes that we had? We have been running all the previous 7 steps by having them still in the tubes. So everything, including amplification and the whole process, we had maintained them to be in separate tubes. Now we're going to take the mixture from these tubes, and then we're going to pass it to this medium, whether it's going to be capillary electrophoresis or the gel electrophoresis, okay? The difference between capillary and gel is going to deal with the 4 tubes again. So it will have kind of 4 channels, 1, 2, 3, 4. Here we're going to deal with one single tube, which you can imagine now it links more to the discussion from the previous class, where we can shoot some laser and indicate the molecule. This is the technology for that. But in the beginning, the gel electrophoresis was being used. And so there is this gel. There are electrodes in the design of this gel electrophoresis. Now, why do we have electrodes? Because we're going to have positive and negative side. Think about a battery, right? We need to move something in between, okay? A positive anode and a negative cathode electrode are connected to that gel medium. In between, there is a buffer solution that facilitates the electric current that's going to go. And so, as this process that's going to see in the next slides animated, as the samples come and go through, that is the DNA sample or any kind of sample such as RNA or protein, there is an electric field, okay? Now, why that electric field makes sense to be there, okay? Can we characterize the DNA molecules by any kind of specific charge? That is the question, okay? In other words, if we have positive and negative medium here for us, or electrodes, how's that going to impact the sequencing? Okay, think about it for a moment. Okay, now DNA, it has an inherent charge that comes from the phosphate backbone of the DNA molecule. Okay, that is a negative charge, okay? So if we want to characterize the DNA through the phosphate, we can refer to it as a negative. So if I pour something from one side of the gel electrophoresis, the molecule is going to travel to the other side. So let's see this in action. We're going to bring these tubes. We'll take the sample from each, put it here into the gel electrophoresis. So we have negative. The DNA itself is characterized by an inherent negative charge. Now, as this sample comes here, it's going to travel towards the positive one. Yet, remember, we had short and long ones. If I take you to this slide here, remember this part, right? We have fragments of different varying sizes. In this case, as those go into here, the smaller ones will travel faster to here. And this speed or this movement affected by the fact that the extra these nucleotides with larger length, okay, is going to move slower in this case through the channel that they will go through. Okay, it's just giving the fact that, you know, we have one molecule that is easier to move as compared to having 3 or 4 molecules attached to other, as an intuition. Okay, so this is the beginning. We are just starting the process. We are feeding it the sample and it keeps going. We can see now, okay, this is kind of longer one. Now the smaller one, this is just to indicate for you they are of varying sizes. Okay, as they keep going, this is what's going to happen as we watch in this animation. Yes, smaller ones are going to move faster, and slower ones, they're going to kind of be slow, the longer size fragments. And this is what we end up with. Sorry, guys. Okay, so this is what's going to be the case here as those travel here through the different channels in front of us. Now, again, since the DNA is negatively charged, it moves towards the positive. The larger, longer molecules travel slower in this gel medium. Okay. Now, the large molecules make less progress than smaller molecules, so they hang here. Now, how this can be helpful, again, I'm going to draw a similar example to what I've showed you earlier. Okay, so let's say I have A, T in the first tube, okay, A, T, C, G, A, D, D. Now we know this is where it stops. And then let me do this, A, T, C, G, A, T, and then C, G, G, okay, and then A, C, C, and then A, D, D. So we know that this, since it's very long, could be here, okay, and this is, since it's shorter, it is the one here. So by knowing, okay, the size, by knowing the size, we can tell the position of the fifth of the ADD, the adenine in this case. So in here we know that this is which base. This is, we know that we have here 5 bases. So adenine is the 5th base. Okay, adenine in this case is going to be the 13th base. Okay, so we are focusing on these DD NTPs, and as we jump from here to here, we can build our sequence. So do you see now how we can build our sequence? But it is the same moment. I'm going to ask you one important question, and it is a treasure hunt question here. I know we have many this time, but bear with me. They're important to make sure we understand. And again, we can discuss any of those in the office hour if you wish. Okay, so what do we see here is that these DD NTPs, okay, are going to be the focus of arranging our final DNA. Okay, now, given what we see here, what could be the limitation of Sanger sequencing? What could be the limitation? Any something that comes into your mind because of this discussion? In other words, we are having different reads at this case. All what we are caring about is the DD NTPs. So all what we have done in terms of DNTPs, they are not the focus. Okay, what is the focus is here. Now, given what we have discussed in terms of randomness and varying sizes, what could be the limitation of this technology? Okay, share it with us as we or with me on as part of the treasure hunt activity. So now it comes the Sanger technology here. Sequence is going to start from the bottom. Why from the bottom? Because we know that this is here going to be, this means that the moment the primer and the DNA tag DNA polymerase started the sequencing, it said, okay, there is ADD, and then it stops. So right at the beginning, if it were to do more elongation, it will be longer. So this is not only says it is a shorter sequence or fragment, it also gives me kind of the ordering and the position. So I can use this piece of information to say we have C here because this is C. Next is going to be T. Next is going to be G. Next is going to be A. And we keep going, reading those in order. And we are constructing actually our DNA sequence. This is how we do the construction. So again, remember, we are going from bottom up because we know that's a shorter sequence here. And it's not only shorter because short, it means it has been the first one read, because that is when we had the DD-NTP stop. Otherwise, anywhere the DD-NTP stop later on, and this technology is going to march that this is going to stop here, okay? It's not only longer sequence, but also give me the position of the DD-NTP. This is one that I'm interested with. We're interested in this one, right? As we go and we construct the sequence. And so the different copies, you see now the big overlapping here, and that is very interesting, really a technology and technique to read the DNA sequence. And here we have it. So to summarize this, I'm going to share with you this video as well to make sure that we all hopefully capture the core ideas.
Hey guys, welcome back to Mr. Basics here. Let's talk about Sanger sequencing method. Now, before moving further, this video is divided into 3 parts. The first part explains the principle of the method. The second part is about classical Sanger sequencing that was used widely in 1980s. And the third part is about modern Sanger sequencing that uses fluorescence dyes. Sanger sequencing is based on dideoxynucleotides in DNA polymerizing reaction. Let's understand what are dideoxynucleotides. The nucleotide has 3 components: the nitrogenous base, which can either be A, T, G, or C, the sugar residue, and the phosphate attached with the sugar residue. When all 3 components are present, it's called nucleotide. If the phosphate is absent, then it's called nucleoside. Now watch carefully. If the hydroxyl group at the 2' end of the sugar is removed, then it's called deoxynucleotide. The deoxynucleotides are present in the DNA, and they're used as a substrate by the enzyme DNA polymerase. All the DNA polymerase, whether it's DNA polymerase 1, 2, 3, Taq polymerase, etc., uses DNTPs as their substrate. The hydroxyl group attached at the 3' end plays a very important role in DNA polymerizing reaction. During DNA polymerization, there is a reaction between 3' group and the phosphate of the new DNTP. In 1977, Frederick Sanger had a brilliant idea. If the 3' group of the nucleotide is absent, then there won't be addition of new nucleotides and the reaction would stop. The nucleotide having 3' hydroxyl group absent is called dideoxynucleotide. Di means 2 and deoxy means absence of the hydroxyl group. Let's see classical Sanger sequencing method. The template DNA to be sequenced is divided into 4 tubes. Each tube has a primer that will bind to the template DNA. All the 4 DNTPs, that is the ATP, the TTP, the GTP, and the CTP are present. Along with these, DNA polymerase enzyme is present that will add DNTPs on the template DNA. Now, here comes a very interesting part. This reaction mixture also has one radiolabeled dideoxynucleotide at a very low concentration. What I mean by this is that tube 1 will have dideoxy ATP, tube 2 will have dideoxy TTP, tube 3 will have dideoxy GTP, and tube 4 will have dideoxy CTP at a very low concentration, and all of them will be radiolabeled. Now let's see what happens in tube 1. The DNA polymerase would add dNTPs on the template DNA and carry out normal polymerization reaction. If somehow the dideoxynucleotide is incorporated, then the reaction suddenly stops. The reaction terminates as there is no 3-hydroxyl group for the addition of new dNTPs. The incorporation of dideoxynucleotide and termination of polymerizing reaction is a random process. Hence, the overall mixture will have several fragments that has experienced chain termination. Now, if the length of the fragment is known, then the location of adenine in the DNA can be estimated. The length of the fragments can be obtained by performing polyacrylamide gel electrophoresis and autoradiography. Once the autoradiogram is obtained, we can now have the sequence information of the desired DNA fragment. Okay, the first nucleotide is going to be A. The second nucleotide is A. Third is G. Fourth is C. Fifth is T. Sixth is T. Seventh is A. Eighth is G. Ninth is C. And 10th is again C. Now let's see modern Sanger sequencing method, which is used even today. Now in this method, each of the dideoxynucleotide is attached with a fluorescence dye. The fluorescence dye can either be yellow, green, blue, or red. Instead of having an X-ray film placed on a gel, we'll now have a gel that would give fluorescence when exposed to UV light. As each of the dideoxynucleotide have their own fluorescence, it's now possible to carry out the reaction in one single tube rather than 4 different tubes. The chain termination reaction would result if dideoxynucleotide is incorporated in the DNA. The chain-terminated fluorescence fragments can be separated by polyacrylamide gel electrophoresis in a single well. The more advanced system uses capillary electrophoresis for the separation of fluorescence DNA fragments. These capillaries are further equipped with CCD or charge-coupled device for the detection of fluorescence signals.
So as we have all watched in that short video, there has been a classical way of doing it. This is coming from the original Sanger publication, giving you how they used to do the experiments. They would use that gel with agarose as a medium, and it was a demanding process. There were no colors there. You can think of radio attached molecules and then an X-ray technology to try to get this all kind of black, white, and gray. And of course, it wasn't easy to do or as accurate as compared to the next version. We're going to see some fluorescence dyes. So this used to happen by hand and not really for high throughput, meaning for large sequences and so on and so forth. So as for the automated Sanger sequencing, this is the high throughput way of doing it, also highlighted at the end of that video that we watched. Now you can think of having everything in one tube. This is what's referred to as capillary electrophoresis, okay? So the difference here is adding ddNTPs and NTPs of all flavors into the same test tube as compared to having 4 tubes. And all the mixture is added to one gel-filled capillary tube. And then the capillary is charged and analyzed, analytes migrate as before. Okay, so here we have everything. As the analytes are excited by a laser, okay, now this is an energy of elimination source. So we have this wave, then certain waves are based on the color of that fluorescence dye. Based on that excitement from the laser, was going to be returned is the wavelength indicating the color, something also we highlighted before. So that specific wavelength is eventually going to highlight which sequence we have. And this links more, this is what I've referred to. It could be that when I started the discussion, this is what you assume is going to be happening or the case based on what we discussed in the previous lecture. But again, this is indeed what happens today, but there were certain developments in between. So I'm gonna done here, and then we'll continue next with the Human Genome Project, and that is coming part.